Hi, welcome all. My name is Laurence. I'm a member of the Protein Society Executive Council, and it's my great pleasure to introduce the webinar today. Before handing over to the organisers, I have a few announcements. First, I'd like to invite you to join us at the Protein Society Annual Symposium, which will be on the 19th to the 22nd of July in Boston this year. And we're celebrating the Society's 40th anniversary. You can still register. Please see the information on the Society's website. Next year's symposium will be on the 9th to the 12th of July in San Francisco. Again, you're very welcome to join us there. Lastly, I'd like to highlight the Q1 Journal of the Society, Protein Science. The outstanding editorial team is led by John Curran, with senior editors and editorial board covering a wide range of modern protein science. The journal also provides best paper awards for the first authors who are graduate students or postdocs, and they're invited to give a talk at the annual symposium along with other benefits. I'd like to cover a few logistics before we start. The talks will each be around 20 to 25 minutes long and will be followed by a short Q&A session, which I will field. Hopefully you can see the Q&A tab at the bottom of your screen, so please put your questions there. At the end of the 4 talks, there'll be more time for questions also. So that brings me to the main event. I'd like to thank the organisers and all the speakers for sharing their exciting science with us today. A brief introduction to the organisers, Laurence, Jaboro and Mcgregory Ogbomo. Laurence and Mcgregory are undergraduate students at the University of Benin, Nigeria, and co-founders of a new and exciting organisation, Protein Design for Africa, of which Laurence is president and Mcgregory vice president. So over to you, Laurence and Mcgregory.
Thank you, Laura. Share screen. Good everyone. My name is Laurence. Protein Design for Africa is a non-profit organization dedicated to teaching African students, graduates, and professionals how to leverage artificial intelligence for de novo protein design to address critical local challenges in healthcare and agriculture. Our mission is to train and empower the next generation of African scientists to develop locally driven therapeutics, diagnosis, and biotechnology solutions for neglected regional diseases.
Okay, so now our first web lab milestone is that we recently competed in an adaptive bio IGEM RBX1 competition utilizing the Bindcraft pipeline to engineer novel protein binders. Although our submissions were not initially selected for the competition's laboratory phase, but Dr. Tamuka, through STEAMulater, recognized the potentials of our work and sponsored the experiment testing of 3 of our designs on the Adaptive Bio platform, where we are currently waiting the validation results. And now for our current therapeutic pipeline, for our next milestone, we are launching a project to design the novel therapeutic protein binder targeting Lassa fever, which is a viral disease that currently lacks a cure and is endemic to West Africa. And to advance this initiative, we are actively seeking mentorship for experts in virology, structural biology, and protein engineering, alongside experimental collaborators interested in validating our computationally engineered design. Thank you.
So, introducing our first speaker, Mike Minson. Title of his talk: Beyond Bending: what computational protein design can build. Dr. Mike received a PhD in 2019 in biochemistry from the University of Colorado Boulder, worked for a startup, built a computational binder pipeline that integrates many of the frontier models, and placed forth at the fission design.
Mike, would you like to go?
Yeah, thank you, Laurence and Mcgregory. I appreciate the honor of being invited to speak to you guys today. I'm going to tell you about some of the things that I've been working on with Protein Design for Africa and give you guys an introduction to protein design. And so to do that, let me share my screen with you 1 second. All right. All right, so the title of my talk today, I'm going to talk to you about the application beyond binding, what you can build with computational protein designs. So my name is Mike Minson. I got my PhD in 2019 from CU Boulder in biochemistry. And in the last 5 years, I've been working on antibody discovery and early phase drug development. And now I've switched focus into computational protein designs. So with that, I'm going to assume that if you're with us today, you are a researcher or an aspiring researcher who's eager to accelerate the pace, widen the access, or lower the cost of doing science using machine learning tools in order to tackle health challenges that are facing the African continent. And so I'm going to assume that you guys are rooted in Africa, you're mission driven, so you're working on widening the access to safe and effective therapeutics. You might be new to computation or interested in applying computational tools to your work, and you're application driven. So you really want to translate what you're doing with these computational designs to design effective therapies for, say, like Lassa fever, like Laurence mentioned before. So to do that, we're going to go through this in 4 sections. I'm going to talk briefly about the mindset that will help you get started. Because I think that there's a lot to learn, and oftentimes when you feel the need to learn before you start, it gets in the way. So we'll talk about that. We'll talk about specific applications, what you can build with computational protein design using some concrete examples and how this might actually be applied in the field. And then we'll go over workflow and tools so that you can get oriented to what the tools are and how they work. And then we'll finish up with some principles and some lessons. Okay, so part 1, let's cultivate some mindset for how to think like a researcher. All right, so generally speaking, people say science starts with a hypothesis. And you to kind of demonstrate that, I'm going to use this graphic here, learn, design, build, test, this cycle of how we do science. And now I want to rephrase this because I don't think that science actually starts with a hypothesis. It starts with an idea, a vision, or a dream. And so rephrasing it this way allows you to take something that you're capable of doing now and getting engaged with the process through curiosity and through vision. So the trap is thinking that you need to learn everything before you begin. And so you can actually get started now because vision and imagination are skills that are available to you now. So the advice that I have for you today is to start now and stay consistent, and learning will compound along the way. Okay, the next thing is technical fearlessness. So You're into this new field. It's big, it's bright, it's exciting. It's also kind of scary because there's a lot to learn. Okay, so the first thing is that you can figure it out. Determination is your most important research skill. Failure is required for learning, so don't be afraid of it. It's okay to be wrong. It's okay to learn. It's okay to iterate. All right, and then the next piece of advice is to start now with curating a quality information diet. So go out there, get a good feed on LinkedIn and X and connect with people. Start reading papers. Find ways to get these new papers into your feed and start engaging in the community because that will compound over years. All right, and so then I'm going to leave with this quote from Jensen Huang, who runs the arguably the most valuable company on the planet today. And he often likes to approach problems with a naive or beginner mindset, which is how hard can it be? So how hard can it be? We're going to find out. Okay. So next, let's talk about some applications. What can you build with protein designs? And so we're going to go over some ideas that I have about how I want to apply computational protein design towards therapy. All right, and so we're going to talk about binders as multi-use tools. Binders, in this case, are essentially protein-protein interactions, and they sit upstream of many of the useful tools. So what the binders unlock are a litany of things that are all downstream of these protein-protein interactions. So you can imagine using binders for vaccine immunogens that present the right epitope in the correct orientation, rapid field diagnostics in low resource settings like ELISA or lateral flow assays that will allow you to detect pathogens in patient samples, thermostable therapeutic candidates. This one's the big one that I'm after against novel pathogens. You can think about it as using it for a therapeutic for pathway antagonism or agonism. Think like GLP-1 drugs, but novel. You can use these for targeted degradation of receptors on the surface of cells, and the list goes on. So to kind of illustrate that, we're going to use the example of a viral target like LASV. And so you can take the surface protein of an LASV viral target, the loss of fever GPC trimer, and feed it into a computational protein design or a binder pipeline. And for the example here, I've got RF diffusion, MPNN, and bolts. And the outputs are going to be binders designed towards this target. And you could use these binders for diagnostics. If they're capable of delivering neutralization, then you can get a therapeutic out of it. Or if it binds the target and constrains it in a certain way, or you can fuse it to a vaccine epitope, it can then also be used to display epitopes for novel vaccine methods. Okay, so the key take home is that if you can imagine it, there's a way to build it. All right, now we're going to go over thermostable neutralizing antibodies for LASV. So this is the project that Laurence mentioned. So at Protein Designs for Africa, Lassa fever is an endemic virus and it causes hemorrhagic fever. So the goal would be to generate some sort of neutralizing antibody that is also shelf stable, potentially. And so here on the right, we've got the 6P91PDB structure, which is the spike protein for the surface antigen of LASV. If we feed that through computational protein design, then what we can do is generate binder candidates towards that spike protein. We can then go and mutate the framework residues and maybe the CDR residues of the antibody with like thermo-MPNN, which is a tool for selecting for mutations that are thermal stable. And then we could refold, rescore, and filter these designs using boltz2 or ESM fold. And then that will present us with some sort of novel binder design that will help for the creation of a therapeutic candidate. So then we can go and test and make these antibodies in the lab, test them for binding to the target. And if they bind the target, then that can create a diagnostic. We could use that for a lateral flow assay, for example. If the further testing of the binder is that it shows that we can get neutralization out of it, then it becomes a therapeutic candidate. And we can also use the process of creating binders to various surfaces and testing in these neutralization tests to map the epitopes that are actually going to be responsible for generating a new vaccine. So you can kind of contribute to all of these activities using computational protein design. All right, so now we're going to jump to the tools. A lot of these tools are open source, which is amazing. So that way you can actually download them from GitHub, use them on your local computer, and have access to these tools. All right, so we're going to break this down to a very simple setup. And so the very first type of tool that's out there for protein design is a backbone generation tool. And what it inputs is if you either have a target, you ask it to generate something from nothing, and what you get out is a backbone. It's a structure. From there, if you give it a structure, another tool would be a sequence design tool, where if you give it a structure, it will give you a sequence that might fit that structure. And then there are structure prediction tools that have all sorts of kind of layers to them. And we're going to break this down into two regular breakdowns. So the one is just a single structure prediction. You get a sequence in and it predicts the structure. Think AlphaFold. The other is co-folding. So you give it two sequences in and you get out two structures that are co-folded together. So that's co-folding. Now, when you group and layer these into tools that orchestrate these two things together, you get code design. And so there are tools that will then also do that co-design. And then the whole class of this, of all of these models, these are called just models, and they hold either, neural networks or protein language models that have model weights and inference and runtime compute, and all of these things will then give you designs on the back end. And then tools that take and pick and choose from each of these things and orchestrate them together are called orchestration pipelines. And so there are tools that call models in pipeline format. So the open source tools that fit into these bins that kind of help orient. So for backbone generation, we have RF diffusion, 1 and 2 are out now. Protina, that's an NVIDIA product. For sequence design, MPNN is like the most prominent class. And so you have like protein MPNN and variants, soluble MPNN, ligand MPNN, thermo MPNN, and these have all been trained to do slightly different things. For structure prediction, we have like AlphaFold. Now we're on to AlphaFold 3, AlphaFold 2, and Multimer. ESM Fold 2 just came out, so that's exciting. ESM Fold 2 has got some great promise. It's exceeding some of the benchmarks. We've got the boltz family. So boltz 1 and 2, there's Chai, there's OpenFold. So there's quite a litany of tools to be used. Now, when you start orchestrating these together, built on top of Protina, we have Protina Complexa, and it will do something where you do simultaneous backbone generation and sequence design. boltzgen is the same thing, where it does inference and it does structure prediction simultaneously. And so then all of these models can be orchestrated using tools that will call and do both. And so a good example of this would be like Bindcraft. Okay, so this is probably one of the most important slides. It's going to sound a little silly, but this is really the big unlock. So in order to democratize access to these tools, it actually requires a fair bit of skill with coding at this point. To get over that, you can use coding agents to help you write code and to troubleshoot the friction points of orchestrating these codes. And so, in plain language, if you hold that design goal in your mind, you go through and you can ask the agent to help you build and orchestrate the tools. So to get started, what you ought to do is download and run a coding model of some LLM. So here I use Claude Code personally, but if you're a student and you have access to Gemini, that's a perfect example of something that you should use. So get a hold of Gemini, get a hold of Codex, get a hold of Claude Code, even with the free amount of tokens, and then you can start building scripts that will orchestrate these tools. You can download these things and run them on your computer. The next most important thing is to get a GitHub. So start working with GitHub, start putting your code into GitHub, updating it. This will allow you to package your tools. And you can set these up to either run locally if you have the compute, which is arguably fairly difficult, but can be done with local hardware. If you can get a hold of something that's up to 16 to 32 gigabits of VRAM, you can run Boltz 2 and Boltz Gen, and ESM and Protein MPNN. And so if you can run those tools, then you can then design proteins. You could also do this on Google Colab, or you can use Docker as a way for taking your repo, your organization of your orchestration models, and packaging them into a structure, sending them to a cloud and renting GPU compute for 30 cents to 50 cents per hour. And that's much cheaper than actually buying your own computer to run locally. All right, so now we're going to talk about workflow. So this is going to give you a very simple loop that I use personally to go and design binders. So our workflow follows 4 steps, and I've kind of grouped them. It's actually more like 8 steps, but the idea is all the same. And so first step is understand the biology and selecting your target. So once you have a target in mind, Lassa fever, for example, understanding how this thing works, where it is, what access in the body fluids have to these proteins will help you understand, okay, I need to target this particular portion of the protein. Then you're going to go and search for a structure, and you're going to prepare that structure. That will be the input into your models. So then from that, you input your structure. You can generate hundreds of trajectories, or backbones as they're called. Then you can inverse fold those backbones to populate them with sequences using like MPNN. Then you can refold and score the prediction structures. So you can use like a Boltz 2 or an ESM fold, and it will go and it will take the structure and sequence that you have, see if, repredict the model, score it, give you, and allow you to then filter based on these metrics. So we're going to go through this step by step to make this easy. Okay, so first is the biology. So you're going to have to start with a therapeutic goal. You want to design a neutralizing antibody towards LASV. So you go and you search for LASV spike protein, you look at it and you say, okay, hey, one thing that I noticed is that there's a triangular type of orchestration or structure. So I'm going to go to this laser pointer really quick. Okay, so notice that it's actually like a triangle. That's a trimer. Okay, so viral antigens are trimers. That's interesting. So we now need to know how to prep that. So we're going to take this structure and we're going to try and preserve the native structure so that the things that are on the outside are the things that I can actually access with the design. So we want to block internal structures so that the designs aren't like binding in the wrong place. And we want to like preserve that native structure. Okay, and so then as part of the prep for these PDB, we might need to renumber the structure's chain lengths in order to fit with the model. So that's, one of the activities that goes on. And then we need to figure out where we want the protein binder to bind, and that's called a hotspot. So we're going to define an epitope that we want it to engage. So one of the ways that we can do that is we can look at this structure and we can say, oh, hey, look at that. There's a fab in this structure, co-crystallized with a version of this protein. And so if we zoom in on the contact points of this fab with this viral protein, we can say, oh, hey, look, this residue right here is really important. Asparagine 143. Okay, so that gives us a residue number, and that defines the hotspot for us. And so then we can put that into our next step. So why this matters, the target chain and the hotspots are the design constraints. So you need to input the structure, it needs to be prepared correctly, and you need to define where you want the binder. Okay, so then the next step would be to input this structure sequence. So I've gone and I've prepared this LASV trimer. I've made it a long single chain so that it folds correctly into its native structure, and it hides the internal faces so that I'm not getting any sort of binding on the inside of the protein. So I use BoltzGen. You can use RF diffusion to generate backbones, or BoltzGen, or Proteina complexa, or any other model. You want to generate as many folds as you can. And I've said arbitrarily thousands, but you can use these models to fold, hundreds of designs to your hotspot. And so as it goes through your generator, you get different novel backbone poses of a new binder towards that hotspot. And how this works is it basically will grab the attention of the model and it will say, hey, look here, try to put this design here. So it pins those contact residues and diffuses the backbone around those sites. So you're going to input length of the binder that you want, secondary structure preferences. If you want a VHH, then there's a mode for that in BoltzGen called nanobody anything. And the goal here isn't necessarily to generate the perfect answer up front. It's to generate a library, a large library. So you want many, many designs, and you'll be able to select from those top designs. So then inverse folding or designing sequences is the next step. So you're going to use protein MPNN potentially if you don't have, like if you used RF diffusion, you're not going to have a sequence for that backbone pose. And so you need to populate the backbone pose. You can also use MPNN to change the backbone folds to, or sorry, not the folds, but the amino acid composition so that it's either more soluble or more thermally stable. And so I put right here in quotes. So protein MPNN puts the diversity into each backbone, and you can generate more backbones that way. And so how this works is it reads the backbone pose and it proposes a new sequence that fits onto the sequence. And so then you can generate a ton. It's cheap, it's parallel. MPNN is really fast. You can do this locally on your home computer. And then the new sequences that you've created with MPN, they don't actually necessarily have structures, and so they need to be refolded and repredicted. And so, like I mentioned before, you can use soluble MPNN or thermo MPNN. Using soluble MPNN has been shown to improve the expression of a lot of the proteins, and so a lot of pipelines you'll see actually use that. So like Bindcraft, for example. And so this is kind of an example of output that came out of my code pipeline. I mutated a variant. I got an improvement in my IPSAE score due to mutating with MPNN. And you can see down there at the bottom what those positions were that got mutated. All right, so then I need to go and I need to predict the complex again using another model like ESM folder or Boltz2 metric, like generate the scoring metrics and sort them and then pick the top candidates. And so here's an example of that with this design here for loss of fever. And so I refolded using Boltz2. This is my design spec 18, and here are my scores. Let's see. All right, so to orient, the scoring filters that are generally the most common to get to know and be familiar with are PLDDT. This is a structural confidence score, which tells you how confident the model is that the design is folded correctly. So if it's not folded correctly, it can't be very confident that it is a good binder. But if it is a good fold, it may or may not bind, but that's, your sort of first metric to look at. The next would be your IPAE or your IPTM. These are predicted accuracy scores of the binder target interface. And they're geometry scores only. They don't tell you whether or not the chemistry is correct and whether you're getting correct adhesion. IPSE score, this is one that has kind of crept on the scene after the most recent, or one of the most recent competitions from Adaptive Bio for the Nipah virus. Everybody jumped on IPSE as like the best score metric that'll tell you that your design binds or doesn't bind. And that stands for Interaction Predicted Score from Aligned Errors. So keep an eye on IPSE scores. They're not the end all be all, but they're important. And one of the last things that I do is I always I do a visual check, and you can kind of see here at the interface of this protein, whether or not the loops and the surfaces match. Does the interface look real? Are there big gaps? Are there clashing residues? Any of those are big red flags that will tip me off that the design isn't great. And then I can, either deprioritize that or look for better ones. And so one of the ways to churn through lots of designs and also not favor any one metric by itself is to use what's called a loss function. So you can use loss function scoring to help you filter designs. This is an example of one, and I've included down here sort of a link to a blog from Escalante Bio that uses a very nice loss function that helps. And that one prioritizes IPSE. So go ahead and take a look at that at some point. All right, so lastly, we're going to talk about some design principles. So Laurence and Mcgregory mentioned the RBX1 activity competition that they participated in. I think competitions are wonderful ways to sort of get your feet wet using a real problem to kind of iterate through a design process and to learn. So I participated in the RBX1 competition, and one of the main things that I was able to pull out of that competition was this concept of avidity. And you saw that in most of the winning designs in the competition was that there were two binding modes that were utilized that improved the affinity of the finished designs. And so how this works, essentially the take home is that two binding modes tethered together are stronger together. And the way that that works is that when one design tethers, it holds the other design at a higher local concentration, so it allows it to bind. And if that one binds, then the other one can unbind and rebind. And so it gives it more dwell time on the target. And so you get higher local concentration and slower unbinding. So one way that you can accomplish this with your designs is you can split the generator attention by chunking your task into different bits. So RBX1 was a design that had two domains, a ring binding domain and an N-terminal intrinsically disordered region. And so what I did is I actually truncated the target into two distinct regions, an N-terminal region that's just the IDR, and a C-terminal region that's just the ring box protein, the more structured version. And I generated multiple designs to each of those. That allowed me to crank through the design space really quickly, and it also allowed me to focus the generator's attention on just a specific section of the protein rather than on the whole distributed design, which had a lot going on in such a small space. So you can take the resulting two domains, join them together using a linker, a flexible linker. So I used a variety of linkers, and then I used what's called inpainting and MPNN mutagenesis to improve the linker sequence. And when you join those two together, you can get a nice predicted structure that looks like this. And this is actually the design that got me the fourth place at this competition, which I'm very lucky to have gotten. But the concept here is sound, which is basically if you can't get this design out of your generator from one shot, what you can do is break your task up into 2 or 3 shots, piecemeal together the components, and then use in painting or MPNN to improve the unstructured regions of your design and improve the overall design as you piece these things together. All right, and so that's the take-home message is break down the task into chunks. All right, and so now this is our last slide here. I wanted to talk about this LASV project and one of the ideas and updates that I have for this project. And this is called CDR in painting. We're going to use the ideas that we just got from breaking the process down and applying that to LASV. All right, and so what we're going to do is we're going to start by treating the CDRs, which are these complementary determining regions on the VHHs, as displayed peptides on a structural backbone. And so to demonstrate this, if you look very closely at this image, you can see the VHH structure here in orange. Let me grab the highlighter. All right, so here's the VHH structure. Here's the native fold complex trimer of LASV, and I've recolored the CDRs. So this green one here is CDR3, and then you have CDR1 and 2 here as well. And so what you can do is treat these as individual design components. The model attempts to do this, and sometimes it does a good job and sometimes it doesn't. But if you have a really big structure like LASV, which is over 1,000 amino acids, it's a lot of compute and it's a lot to get lost in the noise. And so my first design idea here is to take a short peptide instead of a full VHH and design a binder using a short region, and it should be easier to compute to get stronger interfaces. Then the next idea would be that all of these CDRs can also be treated as sort of fingers or hands grabbing onto the design. And just as we saw before, 2 designs grabbing on are better than 1. And so for CDRs, all three arms grabbing onto the design or onto the target is the best way to get improved affinity. And so our project goal here is design strong peptide binders in paint using CDRs, and then to optimize these linkers for thermal stability and solubility. All right, and then finally, what we want to get to eventually, and we're looking for collaborators, is test the binders towards LASV in a binding assay or a neutralization assay, or both. And so, hang on. That's an open invitation to anybody who's listening. Please get involved with us. We'd love to have collaborators. We'd love to get your input. We'd love to have your excitement drive the project forward. So contact myself or Laurence, and we'll get integrated. So I want to take that time and say thank you, everybody. I want to acknowledge one of my mentors. His name is Michael Holden, who's gotten me into protein design. And I want to thank Protein Designs for Africa and Laurence for contacting me and getting me involved and giving me the honor of presenting. So with that, I'll take any questions.
Great, thank you, Mike. There's one question in the Q&A box, which I'll read out. I've read some of your Substack recently, so thank you very much, Mike. As you had taught, protein MPNN is great at scaffold stabilization, but how do you balance that rigidification with the native flexibility needed for loop-mediated binding? If a design shows a 5 times relative IPTM jump over a dead wild-type baseline, but the absolute score is sub 0.4 because of those loops, is that enough of a signal to justify ordering the gene synthesis?
So I think when you get really large jumps like that, that's always like a red flag. You should be concerned that just some simple, and what MPNN does well as it designs mutations, but when you introduce a lot of mutations, a lot can go wrong and a lot can change. So I would be very concerned that just one metric or maybe all of your metrics just suddenly got magically really, really good. One of the big problems I've seen with protein MPNN is when you solubilize an entire VHH backbone, it'll generate a new structure for you or a new sequence. And then if you go to express it, it doesn't express because the thing doesn't look exactly like a VHH. So there's caveats here. What I would say is if it's not expensive for you to order it, go ahead and test it. If it is, then maybe generate some other designs and see if the same thing reproduces. If you get reproducibility in your process, maybe your score or maybe your actual sequences are different, but the time that it takes to do that gives you some sort of confidence that you've converged on something that is probably real.
Great, thank you. I had a question about the CDR in painting. So you said that you use the three CDRs individually, and then you put the designs together. Do you envisage that those will be three different scaffolds or linked, or will this be the three designs within one scaffold? And if the former, what do the linkers look like? Can you tell us a bit more about that?
Yeah, so this is a proposal more than it is like something I've actually executed on beyond just what I did with RBX1. So for CDRN painting, what you need is both sides of the, desired displayed peptide have linkers on them. And they can be shorter, they can be long. And so you'd actually need to go and generate a ton of designs where you vary the linker length and composition on both sides. The inpainting part will allow you to generate a backbone that would connect those. And then the MPNM mutagenesis would allow you to identify what region or which residues would fit potentially in there. And so then you would need to do that for each CDR. So CDR 1, 2, and 3, and you'd have linker on both sides because it's contained within a loop. And so you'd have the N terminal and C terminal um, backbone inpainting needed to be would be required for that. So long story short, I think it's actually a really tricky challenge, but there are ways to get through this using auto research that I've seen to be effective. But you really need a design that is low enough compute that you can get through the compute process fast enough that auto research actually starts working. Like if it takes 40 minutes to fold one design, that gets a little out of hand. So did I get to all the points of your question, or is there still
Yeah,
some?
you did. Thanks very much. We've got a couple more questions, but maybe we'll reserve them to the end of the
I can answer in
webinar.
chat as well.
Okay, great.
Yeah.
Thank you.
Thank you for your time.
So thanks very much, Mike. So the next speaker is Sergi Roda from Nostrum Biodiscovery, and the title of his talk is Making the World a Better Place Through Protein Design. Over to you, Sergi.
Thank you, Laura. Okay. Yeah, so I'll share the screen so you can see. So yeah, I want to first thank the Protein Society and Protein Design for Africa for inviting me. And I think also Mike's talk was great. And I actually will make all of my slides more understandable. So yeah, so I'm Sergi Roda. I'm the director of protein engineering at Nostrum Biodiscovery. And yeah, I want to show what protein design can do on more of an application level, right? Like now we had like let's send a hands-on or tutorial on how to do it, and now I will show you some results from my side or our side about it. So, well, I think it was already explained a bit, but the idea is that in protein design, we have two things. We can have try to find a certain protein sequence that does a certain protein function, which is what we call either protein bioprospecting or study. Like we want to find, I understand why that sequence does a certain function, or the other way around. We just want a function. We have a goal for achieving with a certain protein, but we don't have that sequence that does that function, right? And that's what is right now computational protein design. So why do we want to do protein design? No, I mean, as we all know, or probably, I mean, it's quite well known, is that proteins are the functional molecular machines in life. And basically, you can see here the main functions proteins that do on a very global level. So there are, of course, the muscular proteins, so contractile function. There are receptors. There are structural proteins. There are storage, catalysts, so enzymes. There's transport, and then there's, of course, the immune system, which mainly involves what Mike was saying, like protein-protein interactions, so protein binders, right? And yeah, I mean, enzymes are what make life kinetically possible. And I mean, the current tackable protein design problems of interest are mainly involved on making binding proteins, so antibodies, affibodies, mini proteins, peptides, and so on. But also, one other big topic of interest in the protein design field is, of course, enzyme design, right? For green chemistry, sustainable chemistry, and so on and so forth. But okay, so this is more broadly, but why? I mean, it connects also with what Mike was saying, right? So, I mean, we don't have to have a hypothesis, but rather a dream, no? And why you also have to have a passion for having a dream. So, in my case, I mean, why I've been working now almost 8 years on protein design, and why is it my passion? Well, it's because a lot of reasons on the personal level, but basically. One of them is that it connects with the projects I will present. But first is that my family has had a lot of presence of Alzheimer's disease, and I have seen it since a young age, and that's actually what motivated me to pursue a career in biochemistry. Then also one of the big topics that have always impacted me is seeing the plastic waste accumulation on landfill, on the nature, right? Has always been like a driver for me to try to make the world a better place. And then there's, of course, all the harsh effects from a pandemic without effective treatments, as we all learned from the COVID-19 pandemic, right? So studying and designing proteins can help tackle all of these challenges. That's why it's so important, and that's why it's also so important for me. So how do we do protein design, right? So also, I think Mike has covered quite a lot of the topics, but basically the problem is exploring a certain sequence space, which basically is like the amino acids that we want to place for that protein, right? And then we want to computationally assess, or we can do also experimentally, but nowadays computational protein design tries to predict the fitness of that certain protein sequence. And we want to improve, so go into the maximum, right, to the hill, to the top of the protein fitness function of interest, which can be a binder, can be catalyze a certain reaction, can be like be stable at certain pH, and so on and so forth, right? Or can be a multi-function fitness. So this is how the problem is presented, and then there are multiple ways to explore the sequence space. So one way is nowadays with generative AI models, but also one can look at the protein structure or protein sequence and come up with mutations from a chemical intuition. So basically, it means that you can change your certain residues from the protein according to the knowledge you have on chemistry. And of course, this has been now more deprecated due to the appearance of generative AI models, but it has always been also there. And then the fitness function can be represented in many ways. One of them is through AI-based fitness predictors. So it can be, as Mike was explaining, protein structure prediction methods from protein language. Also, protein language models can be used for predicting fitness properties and so on. And then there's, of course, one thing that I am also specialized from my PhD is using physics-based simulations, which is what we call here molecular modeling, MM-based fitness, which can come from docking, molecular dynamics, and these kind of things, which is also shown in this mini GIF here, right? So, well, one of the first problem, then I will go through every case study. So one of the first problems that I want to present to you is the plastic pollution problem. So as we all know, plastic production is not being reduced globally whatsoever. I mean, this is the world plastic production, and only in Europe has been slightly being reduced, but it's still quite a big amount. And almost 90% of the production comes from fossil fuels. So there's not a lot of recycling yet, especially there's not recycling on the bio-based approaches. So yeah, it's still a monumental challenge, and it's due to their high stability, durability, chemical stability, difficulty for their depolymerization, right? So, well, almost a decade ago now, some scientists in Japan found a bacteria that could degrade PET, one of the main polymers of plastic bottles, was discovered, right? And since then, scientists have been discovering new other petases. This kind of enzyme is called petase. Also engineered versions of these enzymes that are more active and stable, and they to basically try to achieve a sustainable circular economy for this type of polymer, right? And then we in our case we took it from an alternative approach. So what we did is explore the concept that we call basically plurizyme, which is basically taking a protein or an enzyme and adding additional artificial active sites, so we can do multiple functions in a single protein scaffold. So you just have one big, or it doesn't have to be very big, but like one single protein scaffold that can do many functions. And basically in this case is a hydrolase function, because PET is a polyester, a polymer, and it can be broken by water molecules, but these basically the enzymes basically catalyze this chemical barrier, right? And well, to do this kind of introduction of a secondary active site on an enzyme or protein, we do it with computational aided rational design, okay? So yeah, I mean, this is basically the protocol. We use physics-based simulation to find through a global exploration many binding sites surrounding, but binding sites not for proteins, but for the substrate, which in this case is the PET polymer. We try to find many PET polymer binding sites on the protein of interest if we want to introduce that activity. Then we look around. Well, we also have built some molecular, like AI-based tools to add this functionality, but basically we add a catalytic triad, which is a type of catalytic site that can break ester bonds. And then we validate also when you do protein design, like after designing or coming up with some idea, you have to validate it, as also Mike was saying. And here we have always been doing it mainly with physics-based simulation. So our Monte Carlo algorithm, that is our proprietary software that is called Pele, and then you can do it with molecular dynamics, no? And yeah, I mean, then I want to show one case study that we did before going into the one that was related to plastic depolymerization. So here we actually took an enzyme from this volcano island, Italy. It was a metagenomic sample. The DNA was extracted, it was sequenced, and this transaminase capable of aminating bulky ketones was discovered. And basically we used this protocol I'm explaining to you, and we found different binding sites, which binding sites are defined by energetically local minima according to this physics-based simulation. And well, here I have a brief video of how this global exploration works. So here you can see the substrate being moved throughout the protein surface of transaminase surface. And basically now it stops because it finds a local minima, a binding site that is now not functionalized, but basically through site-directed mutagenesis, we change the residues that are being highlighted in yellow, and we add the catalytic triad I was mentioning. So basically through this site-directed mutagenesis, we introduce this hydrolase site that now can catalyze hydrolysis and ester hydrolysis, and now basically with another simulation, local exploration, like an induced feed docking, we see if this substrate is being catalytically accommodated into this new functionalized active site, right? And we see that we do. So well, we basically now pass these mutants to experimentally being validated, right? And well, basically the project was a success, and we were able to transform this transaminase enzyme into a plurizyme that could do both hydrolysis and transamination, coming up with an enzyme that is capable of transforming beta ketoesters into beta amino acids, right? And this is very important because actually 40% of pharmaceutical compounds, being small molecules, of course, contain a chiral amine functionality, which means that basically it's not the same having the left hand version of the molecule than having the right hand. One can lead to very like therapeutic purposes, and the other can be detrimental with side effects. And one of the most famous cases of that is the thalidomide, that basically was a drug to treat nausea in pregnant woman, but when they had the racemic mixture, so they had both the left and the right hand, the woman that were taking those pills had their kids with malformations due to having the other side or the other version of the molecule, the mirror image of the molecule, and that, yeah. So it's very important, okay? And basically these enzymes, the transaminases, can produce the enantiomer, so one of the molecule, like the right hand or the left hand, the right one. And yeah, basically we use the enzyme I developed to design a precursor from an anti-diabetic drug, which is called gliptin, the family of gliptin molecules. And well, it's very important because basically almost 500 million adults have diabetes right now or in 2021, and one of them includes myself. I have type 1 diabetes, so I have insulin. I don't need this, but anyway, it's also very important. And well, so then we use the technology and a protocol that I presented and was validated with this transaminase enzyme on a protein that comes from like a parasite of amoebas, and basically it forms pores on the cell membrane, causing a disturbance on the concentration of ions from the outside to the inside, right? Killing the cell. But basically that protein doesn't do any catalytic function. And what we did is basically use the protocol I mentioned to add on this pore that is formed by this porine protein, being an octamer structure, octameric structure, a set of these scissors, so these hydrolysis sites, being capable then to do ester hydrolysis. And since this octamer structure is quite big, we were able to bind PET polymer particles, being able then to cut the PET polymer and being able to degrade it at will, right? And well, then we use actually this technology even further to come up with a bacteria that was reprogrammed to be able to feed from plastic polymer, so PET, micro and nano plastic versions of this polymer, right? And basically we use this technology computationally, but we introduce like these mutations to add hydrolase site on genes from the E. coli. In this case, we did it with a Escherichia coli, the E. coli, and we introduced it on native proteins from these organisms. So basically with CRISPR-Cas9, we added the mutated versions of these normal proteins or normal genes from E. coli, and that allowed us to basically introduce to these normal proteins that do the normal function from E. coli the additional function of being able to depolymerize plastic polymers, basically. So that allowed us then to basically, from the products of depolymerization of PET, that introducing two genes through a plasmid technology, being able to use those degradation processes as carbon source, basically. And that way, since it basically means that it can feed from that plastic polymer, right? And well, we have been now translating this technology to basically add it into desalination plants. So that way we can, like, once we are desalinating water for water consumption on society, we can also clean oceans from micro and nanoplastics, which is very important because actually there's a lot of pollution of this type of plastics in the world. And actually, it has been seen that it can cause a lot of problems in health for men and especially women because they are endocrine disruptors. So they can like behave like hormones, but in a bad way, right? So that's the idea. And yeah, I mean, these technologies, you can use them yourself. We have this GitHub from my user account, which basically is a tool that democratizes this type of design of hydrolyze sites. Okay. And then, well, we move to the next topic. So one is the related to the Alzheimer's disease. And basically, well, you can see that is a very important problem nowadays because also people are like, there's a more aged population globally. And yeah, it's a very important, they call it the next pandemic, right? So, well, basically what we did is collaborate with a set of public institutions from Spain, Barcelona, to develop an antibody that targets the C-terminal fragment of the main protein from Alzheimer's, which is the amyloid beta precursor protein, that basically the one that when it's wrongly cut due to oxidative stress on Alzheimer's, it releases this peptide and it produces the plaques that almost everyone knows, the amyloid plaques. And well, basically now we are doing a project together with Eli Lilly to develop these antibodies to be able to use them for therapeutic purpose in the not so near future, but in the future, right? And yeah, one of again here the tool that we use for this computational protein design is this AI combined with physics-based or molecular modeling pipeline, because we have seen that if you just stick with AI models, there are sometimes a lot of errors, and although you can generate a lot of diversity and try to stick with diversity, maybe you cannot try too many designs because of the cost of experimental validation. So physics-based basically narrows down these designs by more thorough funneling selection of these designs that you generate, right? And yeah, we have this combination to basically predict first generate antibodies, and then we validate them with protein structure prediction, co-folding the antibody with the antigen, and then we dynamically see with both the Monte Carlo algorithm and molecular dynamics if the antibody sticks there, if the binding mode is the one that produces the binding, what's the minima, energetic minima, so that way we can assess a bit and try to infer what is the binding affinity that we should expect, and so on. And yeah, I mean, basically here I also compiled a bit the methods that you can have from inverse folding design, means the ones that Mike said about sequence design. Diffusion models are models for building this antibody backbone. And then we have also sequence-based methods coming from antibody language models. And yeah, I mean, here I think we can skip it because I think Mike explained it way better than me. But yeah, the idea is that after doing this co-folding method and selecting based on PLDDT, IPTM, and so forth, we have this extra layer of performing a physics-based simulation. And as you can see in the GIF, basically we have like an induced fit docking. So we explore how the FAB, the variable part of the antibody, binds and moves around the antigen's binding site, right? The epitope. And well, this is the protocol we have. And then, yeah, we perform a more like from the ones that pass the previous layer, we do this more expensive layer, which is molecular dynamic simulations, but that way we can narrow down the selection to just 5, 10 designs or less. So that's the idea, right? And well, you can do many analyses. You can look at the energetics. You can look at clustering and seeing the main conformations from that simulation. You can also look at how the root mean square deviation. So basically seeing how you differ from the initial pose, which that can tell you if the pose you were passing, the antibody antigen complex you were passing was right or not, and so forth. So yeah, I mean, another also important thing for antibodies is just beyond binding, you have to care about developability parameters. So one thing also Mike was saying is that these things are stable. They can stay very stable throughout their shelf life. That also they don't produce immunogenicity problems. So basically that when you take these things they can cause side effects on, like, activate your immune system, but in a bad manner, let's say. And then there's of course the problem of solubility and not aggregation. So that this antibody sequence, when it's expressed, it produces in a soluble fraction that can be usable, right? And well, we have also been developing tools for predicting solubility, and we're working on the other developability parameters. And finally, the last case study is the viral threat. So we know, of course, all of us, that this is very important. There are a lot of viruses threatening our societies, especially maybe on Africa. We have the Ebola virus, the HIV. So I think that it's something that, yeah, we have to work on it. And I think that a lot of people are passionate and dreaming that these things can be treated in the near future. And I think that's where protein design can make this a reality. No, so in our case, we also use, I think that Mike also explained it, but we use diffusion models combined with MPNN, and then validation again with this extra physics-based layer. And you can do vaccines by focusing on a single conformational epitope, or you can do a vaccine by combining multiple conformational epitopes, right? So that there's these two potential approaches. And well, basically we're working on several projects to try to tackle different vaccines that are especially important in Europe and Spain in our case, because yeah, we have the funding agencies from national source and EU, but yeah, we have been working with the human respiratory syncytial virus, then also the porcine, the PRSV, which is very important for agriculture purposes. It harms like 50k euros per farm in Europe per average, so it's a quite important virus as well, and there are no current conformational basically conformational vaccines for this, so it's a quite important thing to tackle. And yeah, and also the last slide related to technologies is that we are developing a Spanish platform to basically be able to come up with some candidate vaccines for specific pathogen in less than a week, right? So that way, and it would combine all of this I've been showing. So AI tools with molecular modeling, and that way you can come up with just those sequences. And actually this project will be benchmarked with influenza, aviar flu, and streptococcus, so just not sticking to viruses, but also pathogenic bacteria. And yeah, this is one of the main projects. And I think that we have also been proving that we can work on making binders for viral targets with the Nipah virus, which was also the previous one from the RBX1 competition that Mike mentioned, organized by Adaptive Bio, and basically using our technologies, AI, but also keeping this layer of physics-based simulations, we were able to come up with one of the best binders at the novo selection, because there were some binders that were quite close to things that were already public or were already binders to that Nipah virus glycoprotein target. But yeah, this is basically my presentation. So yeah, thank you, Protein Society and Protein Design for Africa for inviting me, and yeah, it has been a pleasure. So happy to answer questions.
Great, thank you very much. Fantastic talk. I can't see any questions in the Q&A, so did anyone from the panel have a question?
Laura, I think there's questions in the webinar chat at
Okay.
2 o'clock that are getting dumped into the chat instead of the
Ah,
Q&A.
okay. Yes. So have these designed proteins undergone in vivo tests, and how did they respond? I'm not sure which part of your talk that
Yeah.
refers to, Sergi.
I guess they mean the ones from here, no? But I mean,
Yeah.
in vivo, not so many. So yeah, I think everything has been mainly validated by in vitro results. I mean, the one that works with the bacteria is in vivo because it's the bacteria cell. But yeah, I don't know which ones he was referring to specifically, but I guess it's these ones.
And the antibodies against the APP,
Yeah.
have those been tested only in vitro?
No, also in mice, actually.
Right. Okay.
Yeah, and in the mouse model for Alzheimer's, which there's people that claim that it's maybe these models, animal models, could be improved. But yeah, it showed a significant improvement. So on the lifespan or life expectancy of those mice and stuff. So I think
Great.
it's so
Thank you very
promising.
much.
Thank you.
Please continue to put questions in the Q&A box, and they might be answered during the other talks or at the end. So on to the next speaker, who is Tamuka Chidyausiku. I hope I pronounced that correctly, from STEAMulater, which is a fantastic name. And the title of his talk is Protein Design for Accessible Science Communication and Inclusive STEAM Education. Over to you.
Thank you so much. Thank you to Protein Society, obviously, for organizing this. Thank you to Protein Design for Africa for the invitation as an expansion of our collaboration together. Oh, I need to see if I can share this as a slide. How come it's not? Let's see. Sorry about that. Let's see. But yeah, thank you. As I put this together, oh, there we go, slideshow. Okay, can you see my slides?
No.
You can see them now, right? Yeah. Okay, perfect.
Perfect.
Sorry about that. But yeah, thank you so much for the invitation. Yes. And changing gears a little bit, because this talk is less tech heavy, less biotech heavy, but I'm so grateful to both the panelists that have spoken before me, Mike and Sergey, for already giving a really great overview of the impact of protein design, particularly from a commercial standpoint, as well as a therapeutic standpoint, and why this is super important. That saves me a prelude for basically explaining why it's also important to make sure that these tools, this technology, this impactful, innovative field is accessible to students from all backgrounds across all fields and domains, which is the reason of my platform, STEAMulater. I will just briefly go ahead and reintroduce myself a little bit. My name is Tamuka Chidyausiku. I'm originally from Zimbabwe, which is in Africa, and I actually started my journey in science interested in proteins, and it completely unlocked a whole new career field for me, as of course, but a whole new lens in which I look at through the world. My PhD was at the University of Washington in David Baker's lab, focusing on de novo protein design of beta sheet proteins. And since I graduated in 2019, I've been in the pharma biotech industry, focusing on drug design. But all throughout that time, throughout my entire career, I guess now we're approaching almost 10 years, which still, early career, mid-career kind of area. But all throughout, it was very obvious to me that as we're seeing this impactful field grow in terms of methods, in terms of resources, and of course, blooming in many different areas, Africa is not really included in a lot of these different areas, and not just students in Africa, but also even here in America, there've been topics about diversity, equity, and inclusion, and whether they go up or down, whether it's the trend or it's no longer the trend, the fact remains the same that the disparity gap of racial disparity gap, as well as gender disparity gap within science is really abysmal, and it's something that needs to be worked on. And there are lots of organizations that have made a focus on this, trying to recruit students, also trying to acquire grant funding to supply some of these tools to students in different areas. So that's awesome. But there's also a retention issue where students who do claim these science majors end up dropping off maybe later on. And so how do we make science more inclusive? And I use science really as that S in STEAM, technology, engineering, and math being STEM, and then A as the STEM. And that's where STEAMulater kind of came up for me. It started off as really community service. As I was talking to students who had not seen a scientist who looks like me throughout my career. I've had high school students come up, to me and ask, what is this career? How do I get in this career? I've never met a scientist, and students from all different backgrounds asking these questions. And so that's where initially this idea of STEAMulater came about, was, okay, I'm talking to my community, and my community doesn't even know, a lot of folks, whether it's my parents, even my sisters, don't even know what a protein is, and starting from basics of, okay, this is what a protein is. These are the molecular machines that operate, throughout biology. They catalyze a lot of functions within biology. They're really the superstars of the molecular world. And so starting from that basis and really realizing that once people get an understanding of this, they're really excited, they're interested, and then formalizing STEAMulater so that it actually provides resources to college-age students and people who might not have thought that they were interested in STEM, now being able to access this even on social media and worldwide. I've written about some of these ideas of, identity is not siloed outside of your work function. As scientists, I'm sure on this call, Protein Society, we know we bring our whole selves oftentimes to, our science. And so one way to encourage a lot of students to find community within STEM, whether it's through organizations like SACNAS, which is Society for Advancement of Chicanos and Native Americans, who have obviously been working in the space for a really long time, or other organizations like ABRCAMS, American Biomedical Research Conference for Minoritized Students, as well as a few other organizations, including some of the groups here, Protein Society. Finding that community is not easy, but once you do find the community, it is one way to stay steadfast within your studies and stay, until matriculation and graduation, which is, again, one of these big problems within retention where students drop off. And so I decided to also take it a step further, use protein design specifically, since it is the intersection of STEM plus arts. There is definitely an artistic element. Obviously, we've been talking about all these different tech tools that are grounded within the algorithms and the math systems that write them. And obviously, there's an inherent protein engineering element. And so it really combines all five fields in a really nice way. It's the intersection of all of these. And so I decided to use this because also proteins, while we have been talking about them in a therapeutic context, and Sergey talked about them a little bit in the agricultural fields, they really mediate across multiple industries, pharmaceuticals, cosmetics, food and beverage, across different industries and making impact. I don't need to tell people at Protein Society how important proteins are, but when you talk to people for the first time who are a lot of the times have not really realized that it's the same protein that is a big topic in the media about taking supplements and all kinds of stuff, that same idea of that molecule, or that protein molecule, really impacts many different areas. And understanding it is the first step, I think, in a journey to understanding also protein design, of course. And then, as I mentioned, protein design has been making marked fields, marked leaps of advancements, not just, in the educational realm, but also in therapeutics spaces. This I'm showing right here from the Neil King's lab, the first approved de novo designed medicine. Our world is changing as we speak, as these tools are getting, better and better. The impact is being shown across different spectra. So it's no longer this idea of protein design on a de novo perspective is no longer a theoretical academic exercise. It's really showing marked impact. And that impact is not just in the therapeutic context, but also in a commercial sense. And so there is a question about if let's say, since we're Protein Design for Africa is hosting, if African scientists are not at the helm of some of these de novo designed medicines, commercially, what does that do to some of those communities that are left behind? And so it really is important to get everybody from every different background, at least on the board and enthusiastic about this. Obviously, everybody knows about the Nobel Prize, and so it has been recognized even on an international scale. So it has been easier to talk about this in protein design, about why this is important, not only from a generative protein design standpoint, but even in a structure prediction and how that all ties in into us having a good understanding of these molecules and designing the next generation of molecules. For STEAMulater, my main three pillars, and thank you so much, Laura, for the compliment for the name. The name really is designed to obviously invoke STEAM as STEAM education, but also to stir up the conversation in a unique way. As a sole proprietorship organization, it's just me. This started off as community service and then became a platform that was connecting with more and more students. I cannot, connect with massive amounts of college students as the larger organizations do and focus on recruitment. Really, what I could do is focus in some of the innovative spaces, connecting some of the exciting things that are in the pop culture world to protein design and try to introduce students in that way. And then also make sure that the students who are already declared their majors as biochemistry or as biology or oftentimes chemistry find tools that connect with what they already know. So basically meeting students where they're at. A lot of students, including myself, during my grad school journey, whether you're in the lab mixing and pipetting, listening to hip hop music, and really it's one of the most popular genres. And in fact, pop culture, by definition, popular culture really attracts the most folks. And so one thing I would do quite often is, literally zone into the lyrics of a lot of these musicians who are talking about striving, talking about pushing on, talking about continuing on. Because if we're being honest, as scientists, science is hard, and it does take commitment, and does take a way to to further along. And so that's one element that I basically learned from my journey that I am now putting into STEAMulater. And really, ultimately, there are three pillars that I deliver this in. One is what we call molecular minutes. And molecular minutes is basically taking proteins as celebrities, introducing these celebrities to the world. And we discuss about, at this point, the PDB has access to about 350,000 structures or more. And with the advent of AlphaFold, ESMFold, we're not even just limited to just structures or experimentally defined structures of proteins to explain why proteins are important and why the shape of these proteins is important and why it's tied to the function. And so I basically made an app that allows us to highlight protein of the day every single day. It's daily protein, and this app is connected to this larger content machine that makes reels, these short 1 minute to a minute and a half explainers of these proteins. Why are these proteins important? People are, I've had some responses where people are learning for the first time. For instance, Sergi mentioned diabetes, that insulin is actually really important in diabetes. What does insulin look like? And they're looking at insulin for the very first time. And sometimes it's really exciting. This is also a 3D web app, and so people are able to go in, do drag and rotate, start also learning about what are the elements that make up protein structure, really even in some of the nitty-gritty senses. We talk about helices and beta sheets in a very technical jargon way. This is a way for the public to really interact with these elements and learn in a way that is really kind of important to them. I've seen fluctuations in video views and impressions where some proteins really have high interest, some proteins not less interested. And so one thing that I love is the idea of somebody coming in because they're very interested in this protein because it's connected to a specific area that they have highlighted, and they're sort of coming into a funnel where now they're learning about loops and helices and what makes a good protein fold and a good hydrophobic core. And that is a path for them to even stay within protein design. A second offering that we give here at STEAMulater is games. So games that are designed to introduce not just these elements, like break them down even further to the building blocks of proteins, right? So amino acids, and break them down into multiple different ways. STEAMulater Games was designed initially as a social media experience, usually about 3 weeks of intensive hip hop biochemistry knowledge, where I'm showing slides, but I'm also talking to people on either on podcasts or live streams, and people are coming in with their homework problems, but the students are also competing almost in a protein design fashion. To explain this a little bit more, I have a different app that's also on STEAMulater Games. This is one called STEAMulater.com/play, where they're learning about these building blocks, right? Why are hydrophobic amino acids different from hydrophilic amino acids? They're color coded differently. Where in the polypeptide chain of an example of a protein like insulin, or even the whole list of different proteins that people can look at, why are these different? Why is proline special? Why is glycine special? And they sort of color in these amino acids, and using color almost as a heuristic for differences of amino acids. This was we've now had 5 seasons of STEAMulater Games. So this was really early on, where people were learning these basics and trying to get an understanding of the building blocks that make up proteins. And then, of course, we have our, protein amino acid language, where, alanine is hard coded to A, and proline to P, and so forth and so forth. They start also learning some of those single code mechanisms. And it's a way to remember, especially for the students. A lot of my students are also college students or community college students, to remember for their exams, Biochemistry 1 or Organic Chem. It's a way to, come in at a low energy level. And then hip hop biochemistry, which is a fusion of both STEAMulater Games and, more specifically, tying in some of the cultural elements that exist within that pop culture space. I think a lot of students find it corny when when scientists, try to, either rap, or try to engage on a different level. So one way we wanted to do it was really incorporate some of the hip hop aficionados, So I've been a guest on Daily Cannon show with Nick Cannon, where I've been a science resident scientist on his show. This was a show hosted by Amazon, and with Amazon eventually spun off into my own little show, where I explain different topics from a scientific standpoint. And students would come in, they would call in and have questions, and would interact on there. And then also worked with PBS, with NovaStream. How can we make streaming this informal education platform? So this was an escape room where 3 of us scientists were tasked with science-related questions while being on live stream. And part of the challenge was our live stream audience was actually helping us get through this escape room by learning the different things that are associated. My main area, of course, was protein engineering, chemical engineering on a larger scale. And then the only thing second to doing science is really talking about your science. I remember when I was a student at Claflin University, undergrad, the first time I presented a poster of my research was really when I really connected the dots about how impactful what I've just done is, but also how impactful it is to translate that to somebody who might not be familiar to what I am doing. Science communication is the bedrock of a lot of what we do in science, whether it's trying to get funding through grant writing, or you are trying to do outreach, again, activities that we do in many ways. Science communication is something that is important. And so we've sponsored students who have gone through sort of our boot camp of STEAMulater Games, where they learn about all these different elements, and they try to make their own proteins from a natural language processing sort of standpoint. And so we have sponsored them at this international conference, ABRACAMS, usually hosts like 3,000 to 5,000 students from all different backgrounds, and they even have an international element, which is a virtual presentation. And so students, I've sponsored students in Ghana as well as Rwanda to be able to present their research that they're doing at a local university and present it as a poster and be judged. We've even had some winners in previous years. These are two students, Damari Kamara and Ama Nicole, presenting in 2023. And so as a combination of all of these different elements, one thing that I'm very proud to say we've had since the beginning of STEAMulater Games in 2023 is we've had 70 unique students sign up for this, and these are across different 4 countries: America, Rwanda, Zimbabwe, as well as Ghana. And these students have had varied backgrounds. Our audience is college-age students, so 18 and above. Some of them have been stay-at-home moms. Some of them have been community college students. Some of them have been university students. Some of them have been aspiring live streamers and DJs with different levels of really scientific background. Some had little to no scientific background. Most had little to no scientific background, but were engaged throughout this process, which was really exciting. And so in each season, we would have a cohort of students competing for a grand prize. And throughout this, they're tasked with different challenges, almost Survivor style. And so they are voting each other off in order to get that final prize, but you can get immunity to stay in the competition, not be voted off, by answering some of these STEAM-based questions and by designing molecules bit by bit. We've collaborated even with some biotech companies that have really easy access to software. And so I'm really grateful to a lot of companies that are offering some of the credits so that students can be able to use it. One such company is here, Levitate Bio, that we partnered in our season 4, where students now are taking all that theory. Okay, these amino acids form this polypeptide, and we've done this in a fun and unique way by using poetry or lyrics. But now can we look at sequences of known proteins, and then how do those sequences actually fold into a structure, and a really great structure viewer, and then even start playing with some of these cutting edge tools, like BoltzGen, as well as Bindcraft and Protein MPNN. Levitate Bio at the time was really structured almost like Tamarind Bio, and so they allowed access to all these different cutting edge tools so that students can start playing around with them. This is season 4, where it was a collaboration with SACNAS, as well as the University of California in San Diego, Claflin students, as well as University of Washington students of that SACNAS group. And so, students got to at least, start making some models and start getting access to these tools. And, as a result of, now being 5 seasons in, we started, thinking, can we actually take this up a notch as the technology is evolving? One thing has been a question in the back of my mind is, can we actually make designs and actually test these designs? Can students have first-hand experience to not only making designs in silico, but actually having some results come back that can be a feedback loop, and ultimately, have claim to an actual protein that exists in the world and not just on the computer screen or phone. And I tried to ground this idea in an academic context. This is from a paper from David Baker in 2016. A lot of things have changed since then, but this idea was that, native proteins sample a really small sample. Direct evolution kind of, grows that sample size a little bit larger. De novo design really exists in where that blue area is, where now the question is, how do you explore that space? You can explore it with physics-based, models. You can explore it with coevolution-based type AI-based models, deep learning models. And what is the variety of those deep learning models? Diffusion versus, large language models. You can really explore the space in different ways. And so I thought, why not explore it in that hip hop a rap way. And so we created a pipeline in STEAMulater games that we actually presented at our camps. This was a few years ago. This is my students, Demar Morgan, as well as Amma Nicole, both at Miami-Dade College, Community College, and this was their first AberCamps poster presentation. But what we did basically was mine through tweets, essentially, that were for a specific rap context. If you're talking about, who are the gatekeepers of rap or the legends that write a lot of rap lyrics, Nicki Minaj came up with one, and a lot of the students were probs at the time as well. And so we decided to take specific tweets that have these rap lyrics encoded. And then we parsed through all these, try to teach about data capture on ground level. What are the specific algorithms that you could use to actually extract some of this stuff and some of this early data analysis tutorials. And then we basically pruned these down to 539 canonical bars, verifying them with actual lyrics that have been published on genius.com. So making sure that those tweets align with actual lyrics that the artist intended. Since we've had some shout outs and interaction as STEAMulater with Nicki Minaj, we thought, this is great. We have license to really start playing around with this. And, who knows, maybe we can start evolving some of these, artists and music artists into research nomenclature and start involving them in these pipelines. But the students were really excited, obviously, because a lot of students can not only tweet this out, but they're actually, saying them out, spitting them out from memory. And so these are lyrics that really resonate with a lot of students. Either they have their captions for graduation or their captions for, really troublesome time. Now they're playing with these lyrics in this idea of taking, really just the words as almost as a polypeptide chain, since they've learned about the amino acids. And so now here's in this third bracket, start teaching about the difference, right? So the reason why alanine is A is not for any biological reason, right? And it's really order of discovery of those amino acids, and it was assigned that A. So there's no reason why, so there's no reason why, those lyrics should translate to proteins. And so we started actually talking about frequency of proteins. And so amino acid frequency in known proteins, and looking at that, comparing that with alphabetic frequency in the English language. And so grounding it to that. And so then we made a map of the highest abundance amino acid versus the highest abundance, letter in the English language. And we call this, concordance sequences. So we translated those lyrics to those concordant sequences where we have potential viability of that folding into an actual protein. And then we didn't want to stop there, since we had access to some of these tools in Proteus Studio, as well as some of these open source tools like Protein MPNN, decided to apply Protein MPNN to those sequences. So now the lyric is really serving as a backbone of your protein. It's a cultural backbone, but you're also biasing, obviously, your sequences to actually fold into something that would fold as a protein. And we used co-folding to do bolts2 folding to basically figure out whether something would fold or not with PLDD cutoffs that were similar to known native proteins. And then finally, we actually, while protein design field is exploding, automation is exploding. And so we actually sent these proteins to Ginkgo for their wet lab cell-free protein expression assay. And what's exciting is, this is the mapping of those proteins, the concordant portion, but we had all 4 categories, and we only sent the ones that passed a certain threshold that we had for our PLDTs. But then we also then took some controls to actually take, representative of all those 4. So there's a representative of the concordance lyrics. We have some that are raw lyrics that are, as really a control. But we also have some of the MPNN designs coming from a few different buckets. So students try to use a few different methods for filtering. And, there's a whole question, I think, both Mike and Sergey even mentioned it, when you are finalizing what designs to actually move forward into your wet lab design, there are multiple ways you can go. And so students first had that firsthand experience of doing that. And the results that we have from this cell-free assay were quite surprising. First, any result would have been great because this would have been these students who were in the season 5 enrolled. This was their first data set, wet lab data set that they have access to. So they're learning about what cell-free expression is. Okay, what is the typical expression with cells? And so they're learning about E. coli expression, but then they're learning it in this context of these lyrics that are ingrained in them. But yeah, one thing that we did see. We also added controls. And so some of the controls were scrambling just the words so that it's basically a scrambled protein. And the question there was, is it really the order of the lyric or the sequence that you have that's contributing to the expression, or is it just the composition, amino acid composition? And we had some other controls, GFP, which is a ginkgo control, but we also had this de novo protein. This was my thesis molecule. It's an all beta type protein. So I also wanted that as a positive control of something that's folding that has been formed from a lot of these methods. And you can see those
Tamuka, I think we'll need to wrap up soon. The talk. Thanks.
Okay, I will skip over this, but lastly, you've already heard about RBX1 and with Laurence as well as Mcgregory, we were able to submit these designs. And so these are the final results that we have. We even have a binder from these 7 sequences that we submitted together with Protein Design for Africa. And when you compare them across the 300 designs that were selected, as well as the 9 binders, you can see Mike's design up top here and ours in orange. It's really kind of comparable with some of the mid-tier results. Caveat, this is SPR, not BLI. This is BLI, sorry, for us, and then SPR for the rest of those designs. But yeah, just big thank you to Protein Design for Africa. They've been great collaborators to work with, very enthusiastic students, Laurence and Mcgregory, and Chris Bahl for introducing us, University of Benin, as well as Hampton University, that are now looking at further studies of this, and Protein Society. Thank you so much.
Great, thank you. Really exciting and inspiring talk. Let's keep the questions till the end and move on to the last speaker. So our last speaker today is Chris Bahl from AI Proteins, and the title of his talk is De Novo Designed Mini Proteins are an ideal modality for solving challenges in Africa.
Great. Thanks so much. Really appreciate the opportunity to share what we're working on. Thanks, Laurence and Mcgregory, for the kind invitation, and also to the Protein Society for partnering. I think the Protein Design for Africa is a very exciting initiative. So the speakers before me have done a really fantastic job. So I want to also just thank everyone for really setting up protein design, explaining how the algorithms work, how you think about designing your experiments, how you engage with other people. Really exciting to see, Tamuka, all the work that you've been doing in that aspect. And so for my story today, I really would like to focus a little bit on this one particular type of protein that you can design called the mini protein. This has been a big focus of mine for a very long time. I was very fortunate to join the Baker Lab about 15 years ago or so now, where I was able to do the first ever de novo design of a mini protein. And that generalizable approach has really been something I've been following for quite a while. And now at AI Proteins, we're using this approach to try and impact the world and do as much good as possible. While we focus on therapeutics, it's not the only thing that you can use with mini proteins. And so first, I'd like to just share a little bit about why mini proteins are so exciting and why I think that they are well suited for solving challenges in Africa. And then give you all a little vignette on what it looks like to design a molecule that can go and be a drug. So we'll walk through the design process a little bit, how you think about designing your experiments to test. Once you get those experiments, how you make decisions on optimizing, and then ultimately prove that your molecule can be a drug. But first off, let's talk about mini proteins. So here I'm showing a picture of a mini protein, but this is just here to represent a broad category of proteins that have different shapes and structures. And even the way that we define a mini protein is a little bit up for debate. So I think people talk about mini binders, and I think the size is starting to creep up a little bit about what people often refer to as a mini binder. But for us, at least, a mini protein or a mini binder, these are peptides. But they're kind of long peptides. So about 40 to 60-ish amino acids is typically the range that we operate in. And what makes mini proteins different and special is that these peptides adopt a tertiary structure. They fold into a three-dimensional shape, and that shape is what allows them to do all of the cool stuff. And so we need to design that tertiary structure. When we design them, and also the natural mini proteins tend to be very stable. And this is a huge advantage, right? So we don't require refrigeration. They can survive all sorts of harsh treatments that would normally degrade a typical protein biologic. So, antibodies often require refrigeration, and they can't survive harsh chemicals or high temperatures. But de novo mini proteins can do that no problem. And that unlocks applications in areas where refrigeration is maybe not easy to access. And even applications in agriculture where you need a protein to be stable, out in the environment on a farm, and to be able to be manufactured at sufficiently low cost. So that's another really exciting feature of a mini protein. So your typical peptide, if you want to turn it into a drug or an agricultural product, really peptides often struggle with being sufficiently potent and sufficiently stable. And so the way that most people solve this with peptides is through non-canonical amino acids, unnatural chemistry that requires them to be manufactured either through just synthetic chemistry approaches or by using non-canonical amino acids and genetically engineered organisms. But those are still pretty, I would say, frontier technologies that have yet to be scaled up and are still quite costly. But if you can just get away with solving these challenges with using the old-fashioned 20 natural amino acids, then you're very easily can be genetically encoded, and anything that's alive that can make a protein can be engineered to make a mini protein. And so that's one of the things that we really focus on, is using the natural amino acids. And those challenges that people typically use unnatural amino acids for, that's really what the tertiary structure is for in a mini protein. And we get drug-like properties that we specify and control because we're creating these things using generative artificial intelligence. So now when we're thinking about creating molecules to be drugs, and in general, some really important things are target specificity. Does your molecule bind to the target that you are looking to interact with, and nothing that you don't? And with mini proteins, this is actually something that's a bit surprising, and I think is perhaps inherent to the modality, and not something that I appreciated before launching AI proteins and really making binders to a huge number of targets. And that is that I think that specificity is somewhat baked into the modality because we engineer these things to be so durable. One of the ways to be durable is to be highly rigid. And if you're not able to flex, then you don't really have the ability to participate in induced fit binding. So the mini protein, because it's rigid, is more like a key fitting into a lock. And antibodies, right, that bind with loops, they're able to flex. And that ability to be flexible can allow for induced fit and can also allow them to be cross-reactive with different proteins, which makes sense. Antibodies evolved to be part of your immune system, and ideally you would like your immune system to still provide some protection, even as that pathogen, that virus, or whatever you're trying to be immune to, that's going to evolve, right? And so you want your immune system to continue to recognize that pathogen even as that pathogen is evolving. So cross-reactivity, if you're a natural protein, is a feature, not a bug. But for a drug, it's really not the ideal scenario. So you want these things to be very selective. Mini proteins give you that. And we can also make them bind very, very tightly to their targets as well. Immunogenicity is something that's very important. So is your immune system going to interact with the protein? AI models to predict this are getting better and better. We're not going to talk about this today very much, but the take-home message that we've experienced at AI Proteins is that while the AI predictors are getting better, now we have the ability to manipulate these sequences to make sure that the immune system doesn't have a poor reaction. But you need the ability to manipulate these sequences a lot, and natural proteins can struggle with this. And if you have an antibody framework, you can't really change that antibody framework very much to avoid immunogenicity. And if you're only binding with a loop and you have one binding site, the multiple requirements of drug-like properties and activity can be mutually exclusive. But when you build proteins de novo, you can make it whatever you want. You can make as many different starting points as you want. So it's a lot easier to be able to engineer these sequences to avoid the unwanted reaction with the immune system. And then controlling how long the protein stays in the body is another cool feature of an engineered protein, which maybe we'll talk a bit about at the end, as well as this Lego brick modularity. So the ability to bring multiple different molecules together. Okay, so let's dive right into it. So here we're actually going to talk about making a GLP-1 receptor agonist. GLP-1 is probably the most interesting drug target on the planet right now. It's exceptionally valuable. It's helping a lot of people. And there are still some ways to improve upon this. And I think it's a nice test story to really highlight how the drug discovery process works. But before I get into this, I really want to start by acknowledging the team that I get to work with. So here's a picture from our retreat last summer of the AI Proteins team. The story that I'm going to share with you today is really the story of this incredibly smart and dedicated group of people. And so doing science in biotech is very exciting. It's really a team sport. And in particular, the team leader for this is Ben Minan. So here's Ben right here. And so Ben led this efforts, but basically everybody on this screen touched some part of this story and made this molecule happen. Okay, so here is what the structure of GLP-1 receptor looks like. Here we have the multipass transmembrane domain, and we have this extracellular domain. This receptor will receive a signal from the extracellular environment. So here's GLP-1, so this will bind, induce a conformational change that transmits information across the membrane and allows the receptor to recruit binding partners in the intracellular space. Interestingly, there are two receptor pathways, beta-arrestin and G-alpha, or G proteins, hence the name G protein coupled receptor for this family. These have somewhat counteracting activities. So beta-arrestin actually triggers receptor internalization and ultimately blocks the signaling activity of this receptor. When it signals through G proteins, it stimulates the production of intracellular cyclic AMP, and that helps with appetite suppression, triggers insulin secretion, basically the weight loss benefits and the improvement for people with diabetes. So the ideal molecule would signal just through G proteins and not through beta-arrestin. The problem is right now, the semaglutide and the native hormone actually signals well through both of these pathways. So this remains an engineering challenge. So let's look into a little bit more about this. So the GLP-1 hormone itself is a linear peptide hormone, and it's unstructured. So here's an NMR ensemble. It's all sorts of different conformations. When it binds to the receptor, it becomes this helical shape, so it adopts a more rigid structure. So thermodynamically, there is an entropic penalty for this receptor binding to the hormone. But it does bind pretty tightly to the full-length receptor, but pretty weakly to the extracellular domain alone. So about a nanomolar to the full-length receptor and about over a micromolar to the extracellular domain. Now in the literature, it's been hypothesized, and there's some evidence to support this as well, that by binding more tightly to the extracellular domain, you can control the signaling bias for this receptor and trigger G protein signaling while avoiding beta-arrestin signaling. So our design hypothesis was that if we take this hormone and we scaffold it into a mini protein that will rigidify it, pre-organize this peptide in the binding competent conformation. Then that entropic penalty for adopting this shape, we can pay that upon protein folding and not upon receptor binding, and hopefully be able to bind to the extracellular domain more tightly, and therefore bias the signaling. So this is what we're trying to do. We have a fully integrated design, build, test process in-house at AI Proteins. This allows us to move really fast, but it's not the only way to do protein design. I know, as the previous speakers have mentioned, there are companies now that you can collaborate with that will make your designs and do characterization for you. But for us, it's pretty important to be able to iterate rapidly through this. And so we brought the whole thing in-house. So we start the process by designing the proteins that we want to evaluate. Sometimes we'll have high throughput discovery steps that include yeast display libraries. This particular program, we won't talk about that today. We'll individually express, purify, and characterize different designs and then evaluate their activity to see how they behave and what their properties are. And then we'll tune those properties by the binding affinity, the stability, and we do this by designing rationally different substitutions, going and producing another set of designs, and then characterizing them all over again. And so we'll now just track through this whole process. So to start with design, we also use diffusion models. So here, RF diffusion is one of the main ones, but we use basically any and every open source model. We're always grabbing the latest and greatest from the public domain. So it's very exciting to be able to continue to work with the open source community, right? So we initialize with Gaussian noise, and then we slowly denoise until we get designed proteins. And so, what that looks like in particular here is building first a library of backbones that kind of have the general shape that we want, where we're scaffolding this protein. And then we can take the tensor matrices from here and use that when we initialize the diffusion, so we can kind of guide the diffusion so that way we create a globular protein that wraps around the ligand that we want to engage and doesn't really make sort of spurious unfolded connections in binding to the receptor. One of the things to be aware of as you're thinking about doing design yourself is when you're designing your entire protein to interact with the target, sometimes they can create these open or unrealistic looking proteins that may not really fold that well in solution on their own in the absence of target, or even if they do, they may not be very stable in a therapeutic setting where there are proteases floating around in the body that like to chew up open and unstructured proteins. Standard workflow you've seen and described already. So once we have our backbone structure, the next step is to design a sequence for this. And so we do that. We also do iterative steps of adding noise with diffusion models and then denoising a little bit just to kind of wiggle it and redesigning the sequence a few times. That's an approach that we found worked well here, but whether or not that's the best approach will depend on your target and each individual instance. So something to tune. So then once you have designed structures, you want to then filter and evaluate them. And so you want to predict the structure of your design to see, in the presence of the target, to see if it actually will form a complex and the model thinks it will bind as you would expect. So you get a structure prediction as a complex, but you also want to predict the structure on its own to see if it will fold up, if it's predicted to fold up into that shape. Ideally, this oracle step, you want to use a different model than you used for the generative design initially, so that you have independent validation. And we use lots of different scores to then downsample. So you want to create a larger number of designs and then downsample to what you test. In this case, we're using obviously the AlphaFold 2 confidence metrics, but we still use a lot of Rosetta scores as well. Rosetta still has some signal. And so looking at just the free energy of Rosetta, the binding energy, solvent accessible surface area, the interface surface area, and then we have some proprietary algorithms that we are planning to open source later this year. We then selected 96 designs to express, purify, and characterize in our wet lab. And then here's just a summary of those results. So we produced these 96. this work was initially done a few years ago, and we were able to successfully purify about half of the designs. Since then, we've actually purified probably close to 60,000 different proteins at this point. And we've trained a really nice classifier that can predict with 95% accuracy the ability to express and purify these proteins. So this number is much higher these days, but this represents where we were a couple of years ago. Out of these proteins, we also then ran them through a full set of assays. So we checked by SPR, surface plasmon resonance, to make sure that our designs can bind to the extracellular domain. We also checked to see, using a in vitro signaling assay with these receptors, to see if the proteins would actually trigger receptor signaling. And so we did this just at a high concentration, and then we had a number of designs that triggered there. You'll notice that actually there's more receptors that can signal than we saw binding by SPR. And that's, if you remember, basically the natural hormone is present in each of our designs. So if we just did a bad job and this protein's mostly unstructured, it still contains GLP-1, and so it could still signal. So that's why we think that's happening here. We had about a dozen designs that we could express to a good amount, could detect binding by SPR, and showed specific signaling. And then we down selected to one design that we went and further optimized. And so here's what the data package looks like for this design. The internal code name is D5T. Here's the structure bound to the receptor. Here's the structure, predicted structure bound to the ectodomain. Here we're looking at the signaling assay, where if in the presence of a GLP-1 agonist, these cells will express luciferase, and then actually it reconstitutes luciferase. And then they'll start to glow, and when you add a detection reagent. And so you can titrate the concentration of your drug and then see how potent it is. So we're using semaglutide, the clinically approved weight loss drug, as our positive control here. And you can see semaglutide is more potent than our initial hit molecule. Here's single cycle SPR kinetics. So you can see the binding activity is reasonable, triple digit nanomolar right out of the gate with pretty fast off rates. And then here we're using circular dichirism spectroscopy to look at the structure. And so what we're seeing here is a characteristic mixed alpha beta topology, which is what we want. That's what we designed. And as we heat it up, you can see it flexes and then at some point unfolds. So no, it's not the most stable. Just when you heat it above 55 degrees, it unfolds. We probably don't need to be more thermal stable than that to be a drug. But the thermal stability we find is a leading indicator for many other favorable properties. So we went and optimized this molecule. Unfortunately, don't really have time to talk about how we do the optimization today, but I can just summarize and say we make many amino acid substitutions, evaluate how they work, and then combine them together. We also made some substitutions in this molecule in the part that this part of the protein that we weren't really designing in order to improve the stability in blood. And when you do that with a natural amino acid, it actually makes the signaling activity worse. But so here's the optimized molecule. You can see the overall structure hasn't changed. We've gotten much more potent. So now we're just one log away from semaglutide in signaling potency. This actually may be potent enough. So that's still extremely potent. We're single digit nanomolar. But now by the SPR to the ectodomain, now we're single digit nanomolar affinity to the extracellular domain. So very potent binding. Where semaglutide is micromolar for this, we're now single digit nanomolar. So that's looking really good. And we also managed to stabilize the protein in the process. So now we don't actually transition to an unstructured state as you heat it. The protein still flexes, but then it's able to kind of readopt the shape and never fully unfolds. So that's great. So we took this optimized molecule and we wanted to characterize it a little bit more. We talked in the beginning about beta-arrestin activity. So we checked to see, so this is G protein signaling, right? This is kind of the therapeutic activity we're looking for. We also wanted to check in and see the off-target activity that we were trying to avoid. And so this is an assay where we look for beta-arrestin recruitment to the receptor to look for signaling bias. You can see semaglutide very potently recruits beta-arrestin to the receptor, which is what we want to avoid. Ours, we weren't able to calculate an EC50, so maybe there's a little bit of detectable activity at really high concentrations, but we've essentially blocked beta-arrestin recruitment through, even though we're a good agonist. And then the other measurement is, beta-arrestin triggers receptor internalization. So we also checked for that and saw that as well. The receptor was not being triggered to be internalized where semaglutide was causing that. And so really this is the ideal signaling profile here. So semaglutide is a very potent agonist across both receptor pathways. And our molecule is a very potent agonist only for the therapeutically relevant. And we avoid the other pathway that likely triggers side effects. And finally, just in the last minute here, we'll talk about an in vivo study that we performed. So here we took diabetic mice, and these mice are obese and they have a really hard time controlling their blood glucose levels. We fast them for 16 hours. And so they're very, very hungry. And you can inject them with glucose. So you put glucose directly into their blood. This causes their blood glucose levels obviously to spike. Because they're diabetic, they have a really tough time controlling that. If we give them an incretin agonist an hour beforehand, so either semaglutide or our drug, we would then expect that to stimulate insulin release, and these mice should be able to control their blood glucose levels, which we monitor over time. So that's what we're seeing here in this experiment. At sort of time 0 is where we inject the glucose, and if we just give them saline injection and no drug, you can see their blood glucose levels spike really high and they stay high. And if we give them an incretin agonist, they do a much better job of controlling their blood glucose levels. We added the same amount of semaglutide as we did, same number of moles. We have different molecular weights, so we normalize this by number of molecules rather than total mass of drug. Interestingly, it seems like we perhaps in this model have avoided some adverse effects. So here we're monitoring weight change of these animals. This is not metabolic weight change because they're burning fat, and this is actually really acute weight loss because the mice get really sick and they stop eating and drinking water. And so you can see, rapid weight loss over a very short period of time, showing that these mice are just getting sick. And with semaglutide, they get sick much more than if we give them the mini protein. So that kind of nausea effect, we think that by ablating the beta arrestin activity, we're helping with that. And so really the next step is, there's all sorts of other side effects that you get from GLP-1 receptor agonism, including muscle wasting and loss of bone density. And so now you can imagine a polypharmacology where we can link our G protein agonist mini protein to other mini proteins that can do things like help block these other side effects and maybe improve how long the molecule stays in the blood, all these types of things. And while still being just made of natural amino acids, easy to make with bioprocess methods that are more generally accessible, and while also being stable, so it doesn't require refrigeration, for example. Okay, that's the story we got today. Thanks so much, and would be delighted to answer any questions.
Great, thank you, Chris. I'm just looking at the Q&A box. Maybe I can start with a question. Could you just explain again? I think I wasn't paying attention. The rationale for using the mini protein to prevent the beta arrestin signaling. Can
Yes.
you explain a little bit how that the rationale for that?
Yeah, I didn't go into this. And in some ways, it's not very well understood why, right? So this is somewhat of a puzzle. Binding more tightly to the extracellular domain is, for other family Bs, has been indicated in the literature to bias the signaling towards beta arrestin. And the mechanism is unclear. And to be completely honest, I initially went into this project thinking that there was going to be some kind of subtle conformational change in the membrane here, and that conformational change in the membrane would bias towards G proteins or beta arrestin, what the preference is. And that may still be the case. And part of the original rationale was also that if we bind more tightly to the extracellular domain, this would give us more opportunity to kind of futz around with the bits down here that interact with the transmembrane domain so that we could control signaling bias. But as we were reading more and more into the literature, we saw this hypothesis that, well, maybe all you need to do is bind tightly to the ectodomain, and you get that signaling bias for free. And then when we got our initial design, we didn't talk about the beta arrestin characterization for this initial hit, but I can also share that this initial hit also had great signaling bias. So we seem to have avoided the beta arrestin activity right out of the gate. We're still investigating this. We actually have a collaboration with an academic group ongoing right now to try and determine a structure of this drug bound to the receptor so that we can publish and share and hopefully explain a little bit of the mechanism of how this all works, because I think this does seem to be general for the family B GPCRs, and it still, in my mind at least, is somewhat of a mystery in as to how that actually works.
Great, thank you. There's a question in the chat. Is there a computational program suite to predict off-site binding of designed mini binders at other GPCRs?
So the prediction of off-site binding is definitely one we get a lot, and so it's a very astute question. I'm really not aware of any computational method that's able to do proteome scale off-target binding prediction. And even if there was, I'm not sure how accurate it would be, because it's not just a yes, no, it's really about the affinity, right? Zooming out a little bit, right? Every single protein has affinity for every other protein. The question isn't whether it binds or not, it's really how tightly does it bind, right? Two proteins we would say don't bind. If you put them at double-digit millimolar concentration, there's a binding affinity that you can measure, right? Proteins bind to plastic in a tube. Every time you pipette a protein in a plastic pipette tip, that protein is binding to the tube. The affinity is just very weak. So right now, there's no good, accurate computational method for predicting the affinity of a protein-protein interaction. And I think that technology, that capability, is going to be required for a accurate prediction of off-target effects. Because you really want to know not just does it bind to some other protein, but what's the affinity going to be? Is that therapeutically relevant? We're working on developing some methods in the wet lab to try and do that at scale. And hopefully we'll have more to share in the next year or so on those work. But yeah, it's certainly a frontier. And maybe one other thing I can say is we haven't maybe spent a lot of time on this because we also haven't really run into this as a problem yet. So we've made binders to about 150 targets so far. And what we see time and time again is that the binders that we make are very, very selective to the specific target that we were making it for. And we haven't really been able to identify off-target interactions that were deleterious. And we actually always check our binders against a cell to see does it bind to the cell displaying our target protein and not to a negative control cell that has, all a bunch of other stuff on the cell surface, but is lacking our control protein. And that assay, we've always been able to make binders that bind cleanly in that assay. So it's not proteome scale yet, but it's a pretty strong indicator. And then typically we'll go and check to see if it binds to related family members. So other family Bs in this case, or if you're binding another target like a TNF superfamily, you go and find other TNF superfamilies. And there again, we always find selectivity in our binding. And so I think this is maybe a family, a property of the family of mini proteins, that they seem to be more selective than I would have anticipated, given what you would normally expect to find with an antibody. Yeah,
and you intimated that was due to rigidity. Is that correct?
That is my hypothesis. And my best explanation for trying to rationalize why this seems to be a class property, right? If you see it one time, maybe it's that one protein, but we see this time and time again for hundreds of targets with mini proteins that are completely dissimilar. And so my best guess is that it's the rigidity of binding. And there's a little bit of rationale as well, I think, to support this in the antibody literature, right? Oftentimes when you're performing affinity maturation with antibodies, you're not always getting very many mutations in the first shell, the direct amino acids that touch the target. Affinity maturation with antibodies is typically thought of as second shell interactions. And so the idea there in the literature is that the antibody loops are conformationally dynamic, and affinity maturation is removing conformational sampling and biasing those modes toward the binding competent conformation. And so reducing the entropic penalty of binding, but maybe not dramatically altering the enthalpic contributions to binding. And so with that kind of hypothesis being mostly accepted, I would say, not obviously proven in all instances. That those together, I think, is kind of our current thinking. But I would love to explore this more and actually know why. And if anyone out there is a biophysicist and wants to try and figure out why these weird de novo proteins tend to be more selective than we would otherwise expect, I'd be delighted to collaborate.
Another question. By using a hub and spoke model to spin out asset-focused subsidiaries, how do you prevent your core AI engine from losing the diverse proprietary wet lab data loops it needs to keep evolving?
Yes, someone's done their background research here. Yeah, so hub and spoke structures would be delighted to talk at length about business models and biotech. Our strategy really is for the discovery capabilities to be housed at AI proteins. And once you hit development candidate, it's essentially pencils down. You can't really engineer the molecule any further. And that's really the moment where the spin-out company takes it to do the IND enabling studies and ultimately the clinical development. And so that means that all of the data that gets generated on drug-like properties and engineering them, everything that stays within AI proteins. So we don't have to worry about data leaking out into the subsidiaries, and they're really focused. They're clinically oriented entities. So everybody gets to sort of specialize in the thing that they do best.
Great. And one more question in the Q&A. Why do you think Big Pharma in general has been so reluctant to move into the mini protein space? Is it concerns about immunogenicity? Also, could you say a little bit more about what is the state of play in terms of open source immunogenicity prediction?
Yes. So I think immunogenicity is one of them. In general, Big Pharma oftentimes likes to acquire innovation rather than directly innovate themselves. I actually quite literally had a Big Pharma in a meeting tell me directly that they don't innovate, they buy innovation once it's working and are happy to pay a high price tag to do so. Obviously, every pharma is different. There are other pharma companies that are more innovative than others. And I think there's a growing appreciation for the potential of mini proteins, and more big pharma companies are placing bets. I do know a couple of big pharma companies that do have their own internal mini protein capabilities as well. A lot of the big pharmas are starting to use them as tool compounds to investigate targets and understand target biology, and potentially use those tool compounds as drugs at one point. But there are others that are willing to move forward. But I think in general, it's really the role of biotech, small biotech, and startups to take big innovative bets, show the world that it works, and that's really more when big pharma moves in. And as far as immunogenicity goes, one of the tools that I highly recommend is Mix MHC2 Pred, but there are many others. I think the key insight is, instead of trying to predict immunogenicity broadly, is to recognize that we're all slightly different, and we all have our own MHC alleles, and there are many different MHC alleles out there in the global population. And a protein that's immunogenic for one person is not necessarily immunogenic for another. And that's going to depend on your particular set of, your particular genotype, which alleles you have. And so the more accurate immunogenicity predictions, and these are, I recognize, also T cell epitopes, not predicting B cell epitopes. But in general, if you have a strong immunogenic response to a drug, T cells helps to have T cells involved. So you can start with T cells and get pretty far into the problem. And so I would say make sure you're looking at predicting immunogenicity for specific alleles and then extrapolating to populations from there. Yeah.
Great. Were there any questions from the panel?
I had a quick question for Chris first. When you're thinking about designing these mini proteins, what is the impact of the dynamics of your target? I could imagine
Mm.
having a static structure that you're using as your template is great, um, but if the flexibility of the protein and or different conformational changes, are going to play a role, how do you align that with success of the design itself?
Yes, absolutely. Flexible targets are still a frontier for targeted design. What I didn't talk about today actually is that we have an entirely orthogonal approach for doing binder discovery that is something of a fancy new way of doing an old-fashioned idea where we have libraries that are, instead of making a bunch of candidate designs that are designed to interact with one specific target, instead we design a library of binders that are not designed to a specific target. They still have a designed binding site, but we design those binding sites to be as different from one another as we can make them. And if you make enough of them, in our case, we made 1 billion of these different binding sites. There's no randomness, no redundancy. We design out liabilities, so it's an exceptionally high quality library of binders, but it's a naive library. And then you can use a display technology like yeast or phage, pan for binders. And when you do that, you don't have to have any structural knowledge of the target. You just look for binders and then functionally characterize them. And we've actually used that to unlock novel biology. One of these actually is a target, TNF receptor 1 that we pursued. And we seem to have found some novel, potentially allosteric ways of inhibiting that receptor that we're still investigating. But it isn't something we could have used RF diffusion to like design a targeted binder for, because the biology wasn't sufficiently well understood to do that.
Okay, awesome. Thank you.
And can I open up to questions to all of the speakers?
I do have a follow-up question for Sergi. My list. Um, so in your studies where you're looking at bacteria that can, break down plastics, um, can you talk a little bit about the potential toxicity effects on the bacteria itself and how the bacteria may or may not handle them? I'm not sure if it is an issue, but I could imagine if bacteria are taking something in and breaking it down in some way, there must be some byproducts that end up impacting the bacteria that maybe efflux proteins can't handle. And is that going to be the limitation, the survival of the bacteria that needs to carry out this job, going to be the potential impact there?
Yeah, yeah, thanks. It's actually a great question and actually complemented very well our article. So basically, what we saw is like when we added this functionality to the E. coli, it was able to degrade the polymer, but it was eventually not allow like killing it and not growing there. And that's why we added like these extra genes through plasmids to basically be able to feed from the degradation products. And that's actually something very important. Like when you people that focus a lot on this field of degrading polymers, either PET or polyurethane or others, you have to focus on the toxic, like the byproducts. And this is also that's something that connects with drugs. Like drugs, when they go through your body and they are catalyzed by cytochrome 450, like it can be the byproduct that is toxic. So that's one thing that we have to have a clear, a complete depolymerization and a complete removal of the intermediate products, because that's probably the plastic itself is not the problem for endocrine disruption on women and also men, but mainly, yeah, I think it's the intermediate products, the ones that are tricky or the problem, let's say. Yeah, yeah, that's completely true. Yeah.
Mike.
I had a question for you, Chris. So this conversation of drug metabolism just had me, kind of sparked an idea of, now you've designed this mini protein that's super stable. You inject it into the blood. Um, it's probably got a longer half-life, so maybe your, pharmacology is going to change because your dose needs to be, it doesn't have to be quite as high because it lasts longer in the blood. Did you guys characterize pharmacology at all, like half-life in the blood or any of that?
Yeah, important question I didn't talk about today. So actually, because of the molecular weight and hydrodynamic radius, just a monovalent mini protein on its own clears at GFI. So the serum half-life is very short. So, 20-ish minutes. So there are ways to extend the half-life. Semaglutide actually contains an 18-carbon aliphatic chain that's covalently attached to it, and that lipid then allows the drug to bind to albumin, and that extends the half-life. So that's one way. We've done similar things with mini proteins, and you can extend their serum half-life in that way, as well as using the multivalent strategy. So just attaching another domain onto it that binds to something that circulates in the blood. Albumin's the obvious one to do there. Lots of people have done this before, so don't need to reinvent the wheel. But there are many things that you can use to extend serum half-life. So I think that's one of the maybe exciting parts is the ability to tune that serum half-life up and down. And there's instances where you want to go in and diffuse rapidly out of the vasculature and then clear the body very quickly. Radioligand therapy is one of those examples where you're attaching a radionuclide and then using a targeting domain to deliver it to tumors. And that's kind of the ideal pharmacokinetic, pharmacodynamic profile. And there are other cases where maybe you're treating chronic inflammation and you want the molecule to stick around for long periods of time. And so there you might want to maximize serum half-life, have a reservoir in the blood. Maybe you want to hitchhike on red blood cells. I mean, there's all sorts of things that you can do. And so having that modularity to tune the properties to the indication, I think, is a pretty exciting place to be, and not something that I think previous modalities really give you. With an antibody, you've got the FC, and you're going to circulate. According to what the FC is going to do, you can tune it up and down a little bit, but it's pretty limited. Same thing with a small molecule, and so a lot of times you're changing this with dosing, and so that also, with mini proteins, there's nothing working yet for oral administration. We are playing around in this. Mini proteins are a monovalent mini protein is about the same molecular weight as semaglutide, and there are oral formulations for that now. So I think there is the potential there, but it's a very exciting space to think about now that you can somewhat define a target product profile that you want for your drug and then design the molecule that satisfies that rather than figure out what target product profile you can kind of manage depending on the properties that just come with your molecule.
If you limit mini proteins, do you not make them vulnerable to proteases with the linkers? Is there a way to get around that?
Very important question. Standard glycines, polyglycine, serine linkers, the GGS repeats kind of stuff. Those are actually pretty stable in serum. They've been used for a very long time. It's basically every SCFV uses those, and there's quite a few FDA-approved drugs. So, glycine serine linkers are just really hard for proteases to grab onto and cut because there's just not much for them to hold on. But yes, we do need to evaluate serum protease sensitivity. There are high throughput assays. Amgen has some nice papers on thermolysin. That's sort of a proxy for serum, just a promiscuous protease that can be an indicator of that. But ultimately, you just have to get serum and stick your protein in there, see if it gets chewed up, measure it, and see how long you can engineer it. But yeah, so far we haven't had a problem with linkers getting clipped too much. Just been quite nice.
I have a question for Tamuka, about STEAMulater. I think it was great, and the work related to hip hop biochemistry and compiling the bars to try to design projects. I think it was very interesting, and I thought whether using different things from same English language, but instead of bars, I don't know, poems from classic music or classical writings or whatever, or even using other languages would allow you to make better proteins or I think it would be nice. Of course, maybe it doesn't have a full direct therapeutical purpose or whatsoever, but I think that would be very interesting, at least for protein designers out there. At least myself, I think it would be for a nice paper. But I think I would like to see on other languages and stuff. Yeah.
Yeah, nice nice to hear you say you're interested in that function in that area. I think so. it seems to me, obviously, that Protein MPNN is doing the heavy lifting work of actually making any of these top four functional, folding at all. And the fact that, maybe starting from a different language, maybe you might get a different backbone. One thing we did do was comparing some of these different backbones to like fold-seek backbones to see what are we, sampling already naturally occurring backbones? Are we exploring some de novo spaces? I think that's where an interest could lie, is like, oh, maybe you're now looking at, de novo folds completely because you're sampling in this new space and just this new area. But I think bioreasoning is another AI algorithm that came out recently. One thing we'd been discussing with the students at Hampton University was, triaging some of these sequences through bioreasoning and see, do they have any like predicted functions, and then maybe even looking for binding or function based on these. Because right now, yeah, we're just shocked that they express at all, even which is exciting. Yeah.
Very cool. Yeah.
Also, big shout out to Adaptive, by the way, for for doing a lot of the wet lab work on this. Tudor and Danielle, they've been so great about this, and yeah.
Do different languages have different biases of amino acids? I mean, Spanish, German, whatever.
Yeah, I mean, you do see some letters are not used in all if you're just looking at just the ones that use typical it's not typical, but alphanumeric language, and not
Yeah.
even Cyrillic languages, not even
Yeah.
like Mandarin language. If you're just looking at those, some languages, like for instance in Zimbabwe we speak Shona, L is not used in Shona language, X is not
L.
used, so
Right.
we have some students who had even try to do poems in Shona, but because L-leucine is typically integral for a lot of protein cores, and it's not in there. There, it was a question: would that protein fold? Would that be an interesting thing? But obviously, you do a redesign afterwards with MPNN, and so the sequence would have completely changed. So whether the languages themselves are sample different backbones ultimately is, I think, the interesting question there, because we are removing all of that with the sequence redesign. Yeah.
Right. Great, thank you. Well, if we don't have any more questions, then it just remains me to thank everyone for participating in what was a really fascinating webinar, and in particular to the speakers for their exciting talks and the audience for their insightful questions. And with that, I'll close the webinar and hope to see everyone again very soon. Bye.
Thank you.
Thank you so much.
Thank you. Bye.
Bye bye.